So we started the recording, and I think we just need to accept all that it's recorded, so that includes Anders. Okay.
Det er muligt, at jeg ikke ved, hvordan man accepterer. Hvordan ser man, om man selv har fået accepteret det, man skal her?
Det tror jeg, hvis du har ellers kan se og er med, så tror jeg, det er fint
Det er
nok.
fint.
Ja. But I can say until Anders is back that this is
okay. Can you hear me now?
Yeah, now you're back, Anders. Good.
Yeah, okay. I'm not sure how much we actually managed to hear. But
no, it was because we started Stine
recording.
started the video, recording. So just from the top again.
Yeah.
And
okay.
if you share your screen.
I have to share again. Okay. This one. Okay.
Exactly. Yes.
Okay. So let's start again. So this is the first webinar on biodiversity, why it matters and how to measure and monitor it. I don't know if you managed to hear, but my name is Anders Johansen, and I'm a microbial ecologist from Department of Environmental Science at Aarhus University. We have 3 seminars coming planned. The first one is the one we have right now, why biodiversity matters and how to measure and monitor it. Then the next one will be titled Impact of Agricultural Practices on Biodiversity. And that's going to happen at the 14th of January next year. And then we have a third one about valorization of biodiversity in environmental settings. We don't know about the date yet. So, but what do we want to do? We basically want to discuss and be wisened up on biodiversity, why it matters, and how we measure it in soil and among invertebrates. That's today. And also how modern agricultural practices impact biodiversity and what to do about it, if we can do anything about it, to finally how value and business perspectives on biodiversity can make incentives. So it is how to prioritize it in future circular bioeconomy. Okay, the day's program looks like this. I'll just go very briefly, and then I'll first at all I will say that questions for the different presentation, please remember them and put your questions at the last part of the webinar. You can put, of course, comments in the comment track, and Morten will keep a track on them. Okay, but first, from 10 to 10, that's me introducing. That's what I'm doing right now. Then from 10 to 40, we will have Signe Norman giving a presentation. Afterwards, we will have Quentin Geisman giving a presentation, and then we will have a short break if there's time for it, 11:10. And afterwards, we will have Tanasi Saras to talk about modern molecular tools in microbial ecology. And lastly, we will have a short summary and question session. Anyway, so first I will give a very short flyover about CBIO, because it's CBIO webinar that we are presenting. And, you know, Aarhus University have a various a range of centers, scientific centers, and CBIO is one of them. And we are concerned with the circular bioeconomy, as the name indicates. So it's CBIO is a highly interdisciplinary network, and with participation of 8 AU departments and centers. You can see an overview here of the vision that we have, and not least the different disciplines that we are encompassing here. And we are mission-driven and interdisciplinary, and you can see the research and development topics over here in the right. We started out mostly focusing on with strongholds on agricultural research and biomass conversion technologies and so on, but it's been taken further from there. Now we're also encompassing blue biomass and socioeconomics and nature-based solutions. In CBIO, we create value through interdisciplinary research and innovation, partnering up across AU and with external universities. And most important, we also have collaboration and try to have collaboration with industries and other organizations. So the collaboration and multidisciplinary solution drive the shift from fossil-based economy towards a bio-based society with planetary boundaries. So that's kind of our mission. We have a webpage where you can go. It's easy to find. You can see the address, website address on the right side, and that's how it looks when you get there. Anyway, so we're here to talk about biodiversity and why it matters. So biodiversity is important because it's underpinning the ecosystem services that we are depending on. That means it's the services are providing us with food, clean water, medical products, and much more. So you can see here, if this is one way to have an overview of ecosystem services in agricultural settings. So there will be provisional services, regulatory service, habitat services, and cultural services. They encompass different parts of or subgroups of services, ecosystem services, like, for example, a very basic one, providing us with food and clean water, but also giving room for nursery service and gene pool protection. That means that animals and plants can thrive in our environment. So the biodiversity is extremely important in this to be able to deliver the ecosystem services. So most of us know that monocropping is not supporting biodiversity. So that's due to loss of habitat diversity and impacting ecosystem diversity at several levels. So when we are talking about diversity, we can talk about species diversity, and of course the underlying genetic diversity, and all that impacts the ecosystem diversity. And maybe we can hope that diversification of cropping system can revert this development towards depleting our biodiversity, which is one of the big global challenges that we are facing. So here are some photos of more and more diversified agrosystems, also encompassing animals. So this is a figure from a report from EEA, European Environmental Agency, just recently, and it shows very well, I believe, a framework of how biodiversity and kind of fits into circular economy. So there are 3 points that we need to take care of. We need to reduce our resource use. We need to prevent waste and pollution that can happen by reducing or longer use or recycling of our resources. But also, and that's maybe the most important thing in our meeting today, is the biodiversity-friendly sourcing. So that's regenerative farming and resource utilization. So it adopt practices that improve soil health, water and nutrient retention, and native crop diversification. I believe this would be the entrance to the next speaker, Signe.
Yeah,
So,
Signe,
okay. I will stop sharing, and then I would just say that Signe Norman is the next speaker. Signe is a professor at the Aarhus University Department of Biology, and she is also chair of the Danish Biodiversity Council and director for the Center of Sustainable Landscapes under Global Change, Sustainscapes. So we will probably hear something about that. Okay, Signe, yeah. The floor is yours.
Thanks a lot. And I'm just curious to know which, do you see full screen mode?
Yeah, but you just need to start your presentation, I believe.
No, then I'm just duplicating. Now you see it, I guess.
Yeah, but you haven't started, I believe. Press F5.
I have 3 screens, so that's why I'm asking.
Yeah,
because it's on full screen on 2 and not on the 3rd one, which you apparently see. So I'm just stop sharing and share again. So maybe now?
Perfect. Yes, super.
Perfect. So today I will talk to you about biodiversity broadly seen. What is the biodiversity crisis? What does it take if we want to actually change this crisis and recover biodiversity in general? I will take a focus in particular on Denmark and talk to you about some of the main state of biodiversity in Denmark, but also how we can actually move towards actions. So I will start out with talking about the state of biodiversity, what is needed to revert the loss, what will it cost if we should revert this loss? Does increased biodiversity in production systems matter for solving the biodiversity crisis? What are the benefits? So you talked about some of the benefits of biodiversity, but it's very complex to couple biodiversity to actual benefits for humans. And then I round up with some broader perspective and a few perspectives of how we can monitor this. I expect Quentin will talk more about that. And within the Center of Sustainscapes, we work a lot with monitoring. We are monitoring several different areas in Denmark with a dynamic monitoring approach on how we can actually follow biodiversity increase. But action needs to be taken and action needs to be monitored before we will actually see an effect of biodiversity. And you will see that through my talk, and we will come back to that in the end. So what is biodiversity? That has just been said, but biodiversity is the diversity of gene species. It's also the diversity of the individuals within species, and it's the diversity of ecosystems. And then it's very important to understand that it's the interactions among the species and the species with the environments is also an inherent part of biodiversity. Many of you are thinking of biodiversity as just the number of species in a particular place. But biodiversity is not just the number of rare species, it's not just the number of species or individuals, it's the diversity within them and among sites. So if you increase biodiversity in your field with 5 additional species, you have increased the biodiversity there with 5 species, but these species are everywhere else. So you are contributing to a few more individuals or many more individuals, but it doesn't change basically success of populations for surviving over time. So for basically changing the biodiversity crisis, we need to do something about biodiversity in general, have big heterogeneity, assuring that the broad biodiversity of species in Denmark actually can sustain the population sizes into the future. Otherwise, we are not solving the crisis. So yeah,
Signe,
that is a very important
your slide
aspect.
is not
that
changing.
the slide is not changing for some reason. That's very interesting. Yeah. So we are just jumping 2 slides ahead. So you see, I was just talking on this slide. So it's very just important not just to study biodiversity as the number of species, but it's really about functions, their genes, the diversity within and among species, and in particular within and among sites. Yeah. And then I will go through the different slides, and they are all in Danish, but I will talk in English. So now I will go to the state of biodiversity in Denmark. So there's no doubt that the loss of biodiversity in Denmark have not been stopped. And we know that from an analysis of more than 171 indicators, which have been looking at species, habitats and processes. And you see here in green, 1%, where you can say for these 1% of indicators, the loss have been stopped. For most of the indicators, we know for sure that the loss have not been stopped because the condition is too low and the development are not increasing sufficiently to actually sustain these populations and species and indicators into the future. And that is what the figure on the right shows you is in green, the trends which are above sufficient condition for actually sustaining biodiversity. And in red, you can have, if you have insufficient conditions, you can have an increase in number of species, but it doesn't matter in the bigger picture of actually reverting the loss, because you go for basically nothing to something which is still everywhere, so you have not reverted the loss. So if we look at sort of generally how well are Denmark doing in terms of protecting its biodiversity, then this shows a subset of the most important goals that Denmark actually have to fulfill in terms of the biodiversity. And you see a lot of gray and very, very little green. And in short, Denmark has a lot of international goals and targets we have to fulfill. Basically, since the 1979, we have to have the EU Habitat Directives, the Water Frame Directive, and so on. And in general, we are doing very, very bad. We are the second last to last in Denmark in terms of protecting our habitats. So to give you a few examples, if we look to our marine areas, basically, there's 0 where we have actually reached a good ecological state, just in terms of the ecological parameters, but we also have to fulfill the chemical parameters, I've just underlined in red, and we are not even near at thinking about that. So when it comes in terms of assuring good quality of our seas, we are doing very bad. When it comes to good quality of our freshwater systems and rivers, we are obliged basically to revert or basically increase the quality of all rivers and all lakes, and we are far from doing that. It's also below 1% where we have actually reached good ecological state, and for 12% we have a moderate state. And when we then go to the terrestrial habitats in Denmark, it's about 6% of all the terrestrial habitats and freshwater habitats we have to protect in Denmark, which have a good ecological state. So there's still a long way to 100% also for our terrestrial nature. And we are here the second last in Europe in terms of actually reaching this goal of protecting our habitats. If we then look what we are actually doing within our protected habitats, because one of the key aspects of actually reverting the loss is protecting nature or restoring. And when we look at how well we are actually protecting nature within the areas we have designated for protecting, this is the Natura 2000 areas. There's more than 40% of these areas where we have continuous production, both in terms of forestry and agricultural production. You see in this orange color, these about 23%, which is agricultural production within areas which have been designated as protected. And here, again, Denmark is far below the general, or far above the average of agricultural areas within protected areas in Europe. So it's the average is 8 to 11%, and Holland, which we like to compare ourselves with, they have actually below 3% agricultural areas within our protected areas. And we will not manage to revert the loss if we do not stop polluting the natural areas, and if we do not stop actually on a regular basis changing the habitats in forests and fields. So the loss have not been stopped, but what does it actually take if we sort of just zoom out and think of what would it take to stop this loss? It of course requires, as was also said by Anders in the beginning, we have to stop polluting. We have to stop destroying what is left of nature. What is left of nature and the species we heavily have to stop protecting. When we are, to the degree we are still destroying, we need to compensate. And then to revert the loss, it really requires a big effort in terms of creating much bigger protected and restored areas to be able to revert this loss. So often what we talk about is that we try to reduce the pressures of nature, but we are still not, if we don't manage to stop that, we will also not, the efforts we are doing besides that will have to be even bigger to compensate for the potential further losses. So just very overall seen, to recover biodiversity, we need to protect a lot of space, assure that space have quality, and that biodiversity have time in those spaces to actually develop. There's a lot of talk in Denmark of creating more and better nature, but we do not at all have the means at the moment to really do it at a scale where we would also manage to revert the loss. Because what does it take just as a minimum if we want to change that? We have here tried to depict a sort of a Danish landscape with these efforts. So the Biodiversity Council have judged that we need to protect 30% of Denmark's seas, 30% of Denmark's areas, the land at land, and then restore stones and biogene reefs at seas, reduce nutrient for all ecosystems, restore natural water dynamics, assure that was our rivers and lakes actually get into a conditions where it can flow naturally. Water can go from the rivers to the brings, and then there's a big lack of natural dynamics also in terms of grazing dynamics in Denmark. And what is actually the state of protected areas in Denmark today? There is the latest analysis we did says that it's about 2% of Danish land areas can actually can be considered as protected and contribute to restoring the loss of biodiversity, and then up to 7.1%, but that really requires that we are restoring and assuring restoration of these ecosystems. And as many of you most likely know, we got the European Restoration Law went through, and that has some quite large goals for Denmark. And there's also the soil law for soil health. But in terms of restoring biodiversity, it is about mainly about creating large natural areas that are actually protected and restored, and that is the European Restoration Law gives a frame for that, where there's a goal of protecting 20% of European land and seas by 2030, with national binding goals for 2030, 2040, and 2050. And it is so that the countries at these steps actually need to have restored 30% of all the areas that are in bad condition, and we have a lot of land which are degraded. Actually, of the European Commission have made an analysis for Denmark, which is about 2% of the land is in good conditions, and 19.1% is in not good or unknown condition. So together it means that it's about before 2050 we have to have restored about 90% of the land and 60% of the seas. This means that we have to implement effective restoration means on all these areas in Denmark within this time span. So what would all this cost, sort of in terms of thinking, if we want to actually make this happen, what would it cost? And we have made a very recent report in the Danish Biodiversity Council where we have calculated what would it cost if we should assure the minimum conditions for biodiversity, for reverting the loss of biodiversity in Denmark. And what we have done is that we have done an analysis of what it costs, but we have also looked into all subsidy schemes that goes to area-based measurements that has a focus on biodiversity or not a focus on biodiversity. So what this graph shows you in green here, the 207 billions until 2050, shows you that is the number it would cost to revert the loss of biodiversity in Denmark. Unfortunately, we also spend almost 40 billions on subsidies that potentially harm biodiversity. So, and would then increase actually the cost related to reverting the loss. So that is subsidy schemes that goes to continuing farming land, among other things, continuing farming land, which would be better to give back to nature, basically. As soon as there's skewed subsidy schemes in terms of continue polluting, continue farming, instead of balancing the challenges with nature. And then we have looked at a total of 101 billion Danish crowns, gone through all these subsidy schemes, and of those there is 44 billions until 2050 that, if we use them right, can be used to revert the loss. But it's only almost 11 billions that here in dark green that we are sure can actually make that happen. And that leaves a big financing gap in the Danish biodiversity, the effort to revert loss of 162 billions until 2050. And a lot of those, and a big part of this financing gap is lack of financing for actually increasing the quality of our nature and then the natural areas. This is the 56 of this gap is money that are missing for restoration, and there's 30% of the gap that is money that is missing for increased protection of our current habitats, but also the newly created habitats as part of the green tripartite. And if we just look broadly on the ecosystem schemes, there is still financing missing for the marine ecosystem, but there's a big gap when it comes to terrestrial and in particular for the freshwater system. And sort of in terms of how have we evaluated these schemes and what can they contribute to in terms of when we talk about biodiversity. So I focus here in on how does these subsidy schemes and the money associated to them, what is actually the expected effect on biodiversity and land. So what you see here is when to revert the loss of biodiversity, we need to create big protected areas. We cannot revert the loss with efforts outside protected areas, and there we are increasing we can preserve some small habitats, as was shown before with the hares. We can increase some habitats. We can increase the number of common species, but we do not contribute to reverting the loss. And if we look at how the current funding for biodiversity in Denmark is allocated, it's basically the vast majority of the money are going to increasing a little bit of nature outside big protected areas that are restored, and there's basically no money on assuring the quality of that habitat that is created outside natural areas and within the general farming landscape. And you see there's very little that actually creates protection as a restoration and restoration. And if we then look at if now these subsidy schemes would be changed and we would actually implement it, prioritize them towards biodiversity, that requires prioritization of big areas, so that they are contributing to making areas bigger, that they are contributing to increasing the habitat quality of those areas, and that they are permanently protected. So if you remember space, time, and restoration, you will not revert the loss without those. And we generally do subsidy schemes that are 1 or 2 years or 20 years, and it's still not enough. We need subsidy schemes that are 100 or more years actually to be sure that we can contribute to reverting the loss. So I was given the question, does increased biodiversity in the production system actually matter? And I have to, the answer is it doesn't matter in terms of reverting the loss of biodiversity. Maybe it could matter in terms of preserving the genetic diversity of some crops, but then I think you might do something else. It might also matter in terms of increasing the microbial community in the soil. But again, it will increase a quite specific community that might be also outside and in the natural areas. And it's a difference among areas that will be needed to restore to actually revert the loss of biodiversity. So I have tried to sketch it out sort of in terms of where should actions be taken if we want to actually revert the loss. It is, of course, where biodiversity is, where the rare species are, and we need to protect the nature complementary. So that is it could be a long talk about conservation biology, but it doesn't matter to protect the same everywhere. We need to protect the rare and make sure that we within our protected areas are actually preserving these almost 40,000 species we have in Denmark. So again, we need to revert the loss. It's within the big protected areas, and we do not revert the loss when we work outside these protected areas and in the agricultural landscape. So what I have tried to sketch out is in terms of when we go from a field, as we are doing on large scale now within the terms of the biodiversity, the green tripartite, where they will be taken out 10% of Danish area as fields reverting to either production forest, more extensive agriculture, or nature. How will that contribute? So most of these efforts are taking fields, planting them with a production forest, and a production forest is also not providing habitats of long term and a quality that is actually contributing to reverting the loss. And that's the same even if we go to other forest production schemes where we are trying to do it more sustainable. We are doing it a little bit better, in some cases even worse, the science shows. So it is not the efforts that contribute to revert the loss. Here we need to take fields out to restore them. If we take fields out and we revert them to, and as some of the big subsidy schemes are planting with untouched forest, again, we need to assure the real quality of those habitats to actually make a difference. And we are missing a big focus on assuring restoration of existing degraded nature and restoring that. And for those of you interested in the green tripartite, there is the schemes that are most important for biodiversity, they are not in there. So just very briefly, time is running out. We talk often about all these benefits of biodiversity. Biodiversity we need to preserve because of its own rights. For some of us, we get really, really happy when we see a small beetle, which we have not seen in a long time. For others, it doesn't matter. So when we want to try to quantify the economic value of all these benefits, it's very, very difficult. And we have done some of it in the Biodiversity Council, but the challenge is that there's some of these benefits we can actually quantify. When you create a restored protected areas, we have some benefits from creating that area. That is basically protecting the groundwater. That might increase tourism, and it might be better for recreation. It might also be much better for the water environment. So there's a lot of benefits, and it's huge numbers that is related to those benefits. And those benefits are expected to be much bigger than the actual cost that I just showed you. We can just not be sure that we as a society are willing to pay these costs necessarily into the future. Not all those costs that are related to us, our perception of nature and what we are actually willing to pay for that. So if there would have been time, I would have liked to give you a little bit of broader perspective, but I think we have to I will be hanging around to the end, and then we can take it there. Thank you.
Yeah, you're just on time. That's very good. But thank you, Signe. And yes, very nice presentation. We will jump on to the next speaker, and that is Quentin. And while he is loading his presentation, I can say that Quentin is an assistant professor at the Center for Quantitative Genetics and Genomics at Aarhus University. So he is interested in terrestrial invertebrates and their role in ecosystem and how to monitor them with different new technologies, AI and remote sensing and so on. There is a strange phenomenon in your presentation screen. Is it something you did deliberately or what?
I'm not sure. I'm not sure what's the phenomenon.
It's a kind of black
Ripe. Can you see anything at all?
Yes.
Okay. If you try to take it down and share it again, Quentin.
Yeah. Try to restart it. Or maybe Josephine can run it if there is a.
let's try once.
Yeah, let's try you again. Yeah.
How is that?
It's a little bit shaky, but yeah, there's a problem with this. Am I the only one that is seeing this black?
Now maybe
Sounds-
better, but you're not
May-
the only one, Anas. I also see it.
Yeah,
Now
okay,
it's
but
okay.
okay, try to. It kind of disappeared right now, but try to take it away, Quentin, please.
To take away? To
Yeah,
start? you mean?
no, sorry, just start it up.
Okay.
Yeah.
Yeah, thanks for the introduction and sorry for the technical hurdles. Great talk. I seen it before, and yeah, it's fantastic to have more attention on biodiversity. So if we agree that biodiversity has intrinsic or extrinsic value, we still need methods to actually assess biodiversity, which, as was shown before, is actually quite complex. So monitoring biodiversity in space and time is a topic that is not trivial, especially as I will show today for small organisms. So in an ideal world, you would want to take a cube of space and look in time at what's inside. So you would want to count all organisms inside this cube. But also, as was mentioned before, biodiversity is more than just counting species. It's also measuring their interaction, their behavior, processes that they perform. And monitoring organism is hard. A lot of this work is actually performed through manual labor. So this is Helena, postdoc in the lab, sampling in the field and spending many, many hours to just walk on transects and capture pollinators. So because of the high labor cost, our coverage of biodiversity monitoring is sparse in time and space. In addition to sampling in the field and designing campaign, we then need, in the case of invertebrate, especially for insects, identification is actually really hard. So often you would want to have experts to identify specimens. Another aspect of biodiversity monitoring is that it's often for insect destructive. So you sort of put a trap and capture a lot of insects to then study how they are disappearing. So you want to be careful about how much you destroy the environment, especially if you increase scale of your monitoring. And in the last, I would say, 5 years, we've seen methods that are inspired from remote sensing and smart agriculture that fall under what I call the computational entomology, or the use of AI in entomology. And this comes with great expectations. So the idea is to use engineering technique and make, I would say, 2 big categories of tools. The first one would be this is a dream, of course, it's not there yet, but to make a robotic entomologist, so a tool that you could give specimens to and it would identify them. The other category of tools would be the smart insect trap. It would be a device that you can put in the field, and it would be sort of entomology equivalent of a weather station. So it would tell you what, when, and where insects are. But then there is another research part of that would be, if you have these technology, you can actually monitor new things in the field. For instance, observe behavior of insects, which at the moment, the human cost of that is such that it's quite hard to do. So today I'll go a little bit into some specific cases from my group of how we've been developing such technologies, and we'll talk about the promises, but also the limitations that we face so far with these tools to monitor invertebrate. The first case is actually my postdoc work. So it's a camera to monitor insects. And then I'll talk about how we've been developing algorithms to benefit the community. I'll talk about how we've been working on classifying insects better, and how new cameras have been developed in my group to monitor pollinators specifically. And I'll finish with a project on applying these methods to understudied taxa, specifically on worms, earthworms. So the first project I want to talk about is called Sticky Pie. And that was for me a realization that more and more methodologies that rely on remote sensing are used in ecology. In my PhD, I studied insects in the lab, and I wanted to move to the field. And at the same time, as I started my postdoc, there were many new methods to analyze images that became very performant. And that was the whole beginning of AI. So I wanted to learn how to use this method. And the project I began then was simply to use a yellow sticky card. And this is a card, for those who don't know, where insects fly around and they get stuck on a card. This is a common way to monitor insects. And then you would generally retrieve the card and count insects on it, identify insects on it. So the simple modifications was just to put a camera in front of that and to take an image every so often. And the important point of this work was to actually add the temporal dimension. So if we can tell when an insect is captured, we learn more things about insect than we used to. I wanted to create a new method that was affordable, that could work throughout the day. So I ended up using a small computer called Raspberry Pi, and it was powered with a battery. We would also want to monitor humidity and temperature with the same tool. So here you have two different versions of the tool. And on the bottom right, you have an image as seen by the camera. So a large part of the work actually track and identify these insects. So imagine you take a picture every 20 minutes. That's about 500 images per week per devices. By the end of my postdoc, we had 50 devices. So it's actually quite a large amount of data to process. So I developed then a bunch of tools to, I'm not going to go into technical details, but essentially to find insects and classify them. And this has become more common today, where we'd almost think it as trivial, where you can give an image to ChatGPT and it tells you what it is. It's a bit more complicated with insects, because often it's very hard to know what they are, even for an expert from such an image. So this would be a sort of result of processing, just to show you the type of data we'd have. So you would have a time series of insects landing on sticky cards. Sometimes you see there are issues where the lens gets foggy, it rains. So this is more computational entomology in the real world. But yeah, I'm not going to go through the whole video. The important message is, as you see these type of videos, you sort of understand a bit more what happens when insects get trapped. You can see that some actually escape, so there might be biases when you retrieve these cards. You see that sometimes insects get eaten by slugs or by birds. So that also helps, such computational methods helps you to actually correct for such biases. And the thing that I was the most interested about is study the behavior of insects, and in particular their temporal niche. So what I call a temporal niche is, here you have two insect populations. So on the y-axis, this is the number of insects detected per time. And on the x-axis, you have time, where the black bar is the night and the white bar is the day. So showing these two insects, the first one, this is a type of leafhopper. It's going to be active mostly at nightfall, whilst the second one is active in the beginning of the day and in the end of the day, so dawn and dusk. So those are crepuscular behavior, as we call them. So if you classify insects and know where they are, you can actually do that for a whole population and community of insects. And then you can partition this community in space to see how much they overlap. And that's taking the step further from what we do in insect ecology is more anecdotal description of when insects are active. So you'd read an entomology book and it would tell, well, this insect is active at night or during the day, but you can actually now start putting numbers on that. And as was mentioned before, studying biodiversity is more than just counting them. They need, for biodiversity to matter, you need interaction. So insects can interact if they're active at the same time, for instance. So moving on, I wanted to talk about this other project that was done between last year and this year here in Aarhus University. One of the issues with using AI for monitoring biodiversity is it's quite a recent field and everyone reinvent their own tools. So we have now dozens of studies where people take images of insects in different contexts, and then they want to detect these insects on the image. And we waste a lot of effort to recreate basically the same tools. So our efforts, and it's a collaboration between Ecoscience and my center, QGG, was to actually compile all of this data and create a community tool that is open and freely available. So we compiled more than 20 data sets. We created a new algorithm. And the idea is that we can take gigantic or very small images and cut out these insects. So for instance, here are the data sets we used. And with our tool, we can cut them out. And that's an important first stage because you cannot do any sort of analysis unless you actually cut out insects first. So this is a very methodological contribution, but we try to use it as well to study some machine learning principle and learn better how AI algorithms make mistakes when you use them to try to predict when we can use these tools and when we cannot. So moving on now, if given that you have insects that are cut out, you often want to classify insects. So that is to identify insects from an image. And this is a hard problem. Most people have addressed this problem in the community using off-the-shelf tools that have been developed by the AI community. And those are tools that you'd see to classify pedestrians versus car versus this sort of things. And they work fantastically, but in entomology, we actually also use the size of insects. So these tools often don't use the size. Like here, you have two pictures of cats. We don't know how big they are. There is no scale bar, so that's from an entomological perspective, that's an information we wouldn't have on insects. And this is work performed by Melika, PhD student in my group. And here to show you an example, on the top, we have 5 different insects that are shown at relative scale from one another. And you can see that they look fairly similar, but they have quite different sizes. If you were to give this to an algorithm without actually thinking about it, you would give all the images in the bottom. So we would forget the size. So Melika has been working on how to actually feed the size to AI models. And that means AI model could be in principle more performant. They can deal better with some of the problem we face in the community, but also, and that's very important, they become more explainable because now you can tell whether the size was actually useful to classify an insect. And that's something we can understand as ecologists. So again, not going too deep in the technical details. What Melika did is use something related to Bayesian statistics to actually merge the distribution of size of an insect that is prior knowledge with the classification. And what she shows is the full picture is a little bit more complicated, but just as an example here, if she uses a standard tool, and she improves this standard tool with her Bayesian correction on the y-axis, this is the accuracy of our classification tool, and on the bottom is how many images she gave to this tool. She could show an increase in performance, and using that side information is very simple and very computationally efficient. So that's, for me, that's a no-brainer that we should just use this information if available. Moving forward, one of the reasons computational entomology is not yet used at large scale is that most of the cameras we used are actually quite expensive and difficult to deploy at scale. So that's, for instance, on the right, a state-of-the-art camera. So that would be, something like $500 for a camera. And then you still have to buy extra parts if you want a solar panel and so on. So that's actually, if you're thinking of a large monitoring scheme with 100 camera, it's not trivial. Then you have to maintain these tools. So it's hard, and the field, by all means, has not really been moving to cheaper and cheaper devices. Instead, we have sort of an engineer frenzy to add more and more features to these devices. So a lot of them now try to send the data over the internet and to detect insect in real time. And we wanted to take the counterpoint of that and actually make a device that is the simplest and cheapest possible in my group. So Helena has been developing this tool called Minimon, which is a very simple camera, and we focus on having acceptable images from this camera, but many devices instead of a few that make outstanding images. And that's because if you study insects, actually, if you put a trap 20 meters away from another trap, you'll monitor quite different insects. Say the host plant could be different very locally. So Helena has been building this tool, and we've been using it primarily for pollinator monitoring. And once we have this tool, we can record images. For instance, we can take an image every minute and observe the pollinators coming. So this is, for instance, the stream that Helena has been taking. And you can see there are sometimes detection. They could be false positive, and these are from Flatbug, which is the tool I presented earlier to find insect in images. So again, I'm not going to show the entire video, but it's just to show that you can actually start detecting insects over long time series. And once you do that, you can then classify them and understand behavior much better. By the way, those are 3D printed flowers that we use for standardizing pollinator monitoring across different countries and along the season. The last thing I wanted to talk about is a project I call Global Warming. And this is a project where we have 3 people in my group working to monitor earthworms, both in the field, in a lab, but also to classify them. And the whole idea is to take all this work that many collaborators are doing on insects, but to actually study earthworms, which are by all means understudied. So the first part of this project, which is still in its infancy, is to classify earthworms. There are not so many species of earthworms. So in Denmark, you might have something like 20 common species, but they're actually very difficult to identify. So if you look at earthworms, they kind of all look the same. It's not as interesting as insects. So experts actually look at microscopic features on the earthworm, but also macroscopic features that are shown here on the right. And the idea for us would be, can we take an image, a good image of these earthworms, and actually classify them more easily? And that means you could actually sample and identify earthworm at larger scale. So what we've done so far, we started, and this is work by Marta and Dev, PhD students in the group. acquiring a collection of images, again, a collaboration with Ecoscience. And that's here to show you an example of some of the earthworm images we have. And this is what an algorithm would see if we train them to classify earthworm. So the color on top shows unique different species, and those are all different specimens. So we're still working on training algorithm, but the preliminary results are quite promising. So that's work by Marta on a slightly different set of images. But Marta was able to show that she could classify earthworm to a certain accuracy, so 70% accuracy for genus level classification. But it's still something to work on, and mostly now with more images, we should be able to improve that. We want to also improve the algorithms themselves. At the moment, earthworms are we just give an image of an earthworm to an algorithm, but it doesn't actually understand their anatomy. So we could do that more readily. Another thing we're interested in is monitoring earthworm in the field, and that's much harder. So earthworms are obviously cryptic. You would go in the field and you don't even know they're there. So we have been thinking of 3 ways of monitoring their activity. And I'll present just one today, which is the simplest we can think about is a certain functional group of earthworms actually go out mostly at night and they forage on the surface of the soil. So the first thing we're doing is putting cameras over the surface of the soil so we can see earthworm, which is not actually something people do. So we hope to, with such a phenomenological description, we can come up with more interesting hypotheses. So we've been establishing a field trial in AU Vibo, where we put Helena's cameras with red light that doesn't interfere with earthworm behavior. And this is an example of a video that Zix, postdoc in the group, acquired. So this is obviously speed up through the night. But you can see that, so this is about a 70 centimeter square of soil. And you can see how many earthworms are in and how much they move. And that's quite remarkable for us. Is you actually put a camera down and start observing behavior. And it's, even though you read in the literature that there are 50 earthworm per square meter or more, it's hard to believe until you see them move. It's not only just because we want to see them move, but it's knowing there are earthworm and actually observing what they're doing is quite a different thing in terms of monitoring biodiversity. For instance, here you can see them maybe migrating from one field to the other. You can see them foraging litter. Oh, sorry about that. And the last thing we do in this project is that's started by Dev in the lab is to actually monitor earthworm behavior one by one in standard control condition. And then we can study how earthworm learn, how they forage, whether they compete with one another. But this is just started. I just mentioned it for completeness. So in terms of promises and pitfall, what we can actually do today with computational entomology, we can take images and automatically, but it's still very expensive to do so. If you compare, say, just putting an inexpensive trap and recovering it next week versus putting a camera, maintaining the camera, replacing the components and so on. So it's still expensive to do that. We're quite good at detecting insect, classifying insect, tracking insects, but we do very well on data we already have. In fact, often we underperform in new data. As a general rule, if there is a task that you could, teach a child to do in 10 minutes from an image, you can probably teach an algorithm to do that and trust the results. But more than that, for things where you need an actual expert, it's a bit ambitious to think that, hey, I can just solve your problem. There is a bunch of things we can do with with AI that are underexplored, where we can actually do so-called unsupervised methods and discover similarities between specimens or measure quantitative variables like the age of insects and so on. There are a bunch of things we cannot do. As I mentioned, if you have so-called domain shifts, so when you use a new camera and when you have new taxa, when you work in a new field, this AI is often going to fail. And AI is also not built in to know how good it is. So it has very little introspection. Most of these algorithms will be very overconfident. So they will tell you, this is a bumblebee, I'm 99% confident, but they make more than 1% error. AI is also not so good at class imbalance. There are insects that are sometimes more than 1,000 times more abundant than others. So that means you have much fewer of these data to train your algorithms, so they tend to be underestimated. And one thing we cannot do definitely for the foreseeable future is classify all insects at the species level. So often experts cannot classify insects without dissecting them or looking at them under the microscope. It's a little bit arrogant for us to think we could take a small picture and tell what insect they are. So that this is something we need to go over and maybe not try to study specific biodiversity, but actually study other aspects of biodiversity in combination with traditional sampling methods. Yes, and on that, I want to conclude with a take-home message. So cameras and AI are reaching some sort of maturity for insects. They cannot replace good questions and experimental design. They come with their own biases that we need to know and study. And we still need, and we will still need, I would say forever, domain experts and people who work in the field. Thank you very much.
Hi, can you hear me?
Yes, Anders.
Okay, because while most of the talk Quentin gave, I was kind of kicked out of the system for some kind of security reason. But I managed to come back after. Yeah.
Some people started drilling just outside my office, so I hope it was okay. It was quite distracting.
So sorry, I missed most of your presentation, Quentin. Okay, but nobody died, so we will simply carry on. So what time is it now? Maybe it's a good idea to drop the pause and go directly to Thanasis due to all this. Thanasis, are you there?
I am here. Can you hear me?
Yes, we can. Then can you load your...
Hopefully now you can see me as well. Yeah, just give me a second.
Yes. Thanasis has been working with us at the Department of Environmental Science. So he's actually been, what should I say, the pushing force for introducing much of the sequencing technology we have employed. So Thanasis has been involved in a huge amount of projects doing Illumina sequencing and not least Oxford Nanopore sequencing. So unfortunately, he just left us and is now in Novonesis taking care of their strain collection and security, I believe. Yeah. Okay, Thanasis.
Yes,
good to see you.
Yeah. Good to see you, Hannes. Thank you for the introduction. Let me try to share the right screen. That should be this one, right? Can you all see my presentation? Does it look good? Because I'm using a big monitor. Maybe I need to change something.
It's a little bit squeezed from above
it's
and below, I would say. Yeah.
Let me just change the settings real quick. Yeah, sorry for the small delay.
Not a problem.
I think if I do... Is it better now?
It is. It's super.
Yeah, good. We keep it like that.
Yes.
Awesome. So, yes, thank you for giving me the opportunity to talk in this webinar. As Anna said, I was part of Aarhus University until this September, where when I moved to Nova Nessis. So the work I'm gonna present to you here is based on what I was doing for the past 7 years, and has nothing to do with my role at the company. I put that as a disclaimer here. So we started the webinar looking at Denmark and landscape and working with biodiversity. Then we moved over to smaller creatures that we can capture on camera, but there are smaller creatures still that these techniques are kind of impossible. And this is where molecular tools come into place, and my work in the past. Some of you may know some of the points I will raise here, but I'm pretty sure that there will be something for everyone to gain. So without further ado, when we're talking about molecular ecology, we're talking about a subdiscipline of ecology that is concerned with applying molecular genetic techniques to ecological questions. And we can take it a step further, and we can go with molecular microbial ecology when we look at microbes. Of course, we cannot take a picture of them. We cannot see them with the naked eye. So what we do is we extract DNA or RNA, the nucleic acids, from a variety of samples. And the techniques that I will show you here are applicable to no matter what kind of sample you have and what environment you're working with. So here in this picture, I can find a pointer somewhere here. Yes, right there. We have a soil, we have an ice core, we have that's a crystalline hole from the Greenland ice sheet, or a collection of microbes growing on a plate. It doesn't really matter where the samples come from. We employ different sequencing approaches when we extract DNA and RNA, and of course, many of you may know them. Most people are familiar with Illumina sequencing and related ones, and we call those second-generation sequencing, where we get many sequences but short. And then there is the recent advent of third-generation sequencing with PacBio and Nanopore leading the way, where we get, again, many sequences and long reads. And when it comes to sequencing, apart from the platform, we also have the approach. And I make this very general distinction of them in targeted and non-targeted. And we're going to look at amplicon sequencing here in the targeted. We're not going to go into whole genome sequencing, transcriptome or single cell, because they are for specific organisms. And we're going to dive a little bit in metagenomics and metatranscriptomics. So starting with amplicon sequencing, this is a great tool that allows us to perform community level analysis, where we target conserved gene markers, and we answer the question, who is there or who has been there, because DNA is quite stable. The general experimental design is more or less the same, no matter what approach, target, organism one may have, where we start by extracting our nucleic acids, doing depending on the technology, either a 1-step, 2-step, or 3-step PCR with primers, and then some sequencing, and we use bioinformatics tools to analyze our samples. However, something that I would like to make all of you aware, in case you don't already know, is that when we do these kind of analysis, we introduce a lot of biases at each step, and it's very, very important that we are aware of those. So, for instance, how do we collect the sample? What kit do we use to extract DNA? What gene target we are interested in, and specifically, depending on technology limitations, what sub-region of that gene we are analyzing. All those will affect the end result. This is something that we should be aware of, especially when we try to compare our data with published data or previous studies that did not have the exact experimental setup. I'll give you an example here of the 16S gene that most of you know. As you can see in this graph, this shows the standardized entropy, meaning that the lowest it gets, the more conserved this specific region is among a wide variety of gene sequences that have been analyzed. And as you can see, there are different regions, 9 to be specific, 9 variable regions that we can utilize to analyze our communities. And every so often we get these valleys of low entropy where we design targets so we can sequence the region in between. And Illumina sequencing and related short-read sequencing technologies cannot cover the entire gene. So then we have to make an educated choice on which region to focus on. This paper, I have the link here, shows the kind of resolution you get depending on which regions you target. It doesn't really matter what where the taxes sit here. What is important for you to understand is that these trees have the same topology. And of course, the redder it gets, the more unclassified operation taxonomical units you have at species level. And you can definitely see that when you are analyzing all 9 regions, you basically have full species resolution on your samples. But if you try to choose different ones, the red part changes. Of course, with short-read technology, we cannot really do much about it. But this is where third-generation sequencing comes into play. And I have put this paper here. This was published in 2021 when nanopore sequencing was still in its infancy. Quality was not amazing and bioinformatic tools were not really there. But they had already managed to get species-level resolution on human gut microbiota. And of course, since then, this has widely been adopted for environmental samples. However, we still rely on primers that will have inherent biases, as I mentioned before. We also need to populate databases with high-quality sequences. And of course, because third-generation sequencing is a newer technology, we need to develop new bioinformatic tools for our analysis. So then the question is, how do we overcome those issues? And one popular approach is to do metagenome sequencing or shotgun metagenomics, where when we have our population here, we extract DNA. With amplicons, we only get those small sequences of 16S, 18S, IDS, whatever your target is. Whereas with metagenomic sequencing, we basically sequence all the DNA we can get. This will tell us who is there. It will give us answers about all domains of life. There is no primer bias because we don't target specific genes, and we can also see potential functions. Of course, with this kind of approach and DNA-based approaches, we can see what the potential of a community is and not what they are actually doing. And we have implemented that in the past. This is work led by one of our former PhD students, Ate, where we explored microbial diversity. Now, we say in Greenland ice sheet, I have examples from Danish soils, but this is work in progress, so I didn't want to present anything here at this stage. But as I said, the methods are transferable. And what I would like you to appreciate here is that with metagenome sequencing, we can perform analysis like this that shows how much of the entire community is comprised of prokaryotes and how much of eukaryotes, and how it changes in different environments, which is something that you miss when you are going with amplicon sequencing, that you decouple your different communities. As I said, on top of that, you get information about the potential of these communities, and then we went further to investigate that. That might be very relevant, especially when you try to understand ecosystem functions and loss of biodiversity and how it affects other processes as well. Having said that, we still don't know what of these functions is actually expressed. And if one is only interested in knowing who is there, this is still a quite expensive approach, particularly because you have to sequence all of the DNA just to get a little bit of information out of it. This paper from 2018, I really like, suggested that if you do, they call it meta-total RNA sequencing. At the Department of Environmental Science, we call it total RNA metatranscriptomics. There are many terms, but they showed that if you are only interested in analyzing biodiversity, you can go away with sequencing 20 times less compared to shotgun metagenomics. This is very important because all these two approaches require a lot of data, and sequencing, even though costs have dropped, is still quite expensive. But so let's talk about that. We call it total RNA sequencing. It is, again, an untargeted sequencing approach of the entire community, no primers used, no primer bias. And it does cover bacteria, archaea, and eukaryotes. And in the end, what we will get is full-length RNA sequences that we can use for taxonomic identification. And we can also see what functions are actively expressed. The library preparation and sequencing is a little bit different from before, but more or less the same in general terms. And of course, the data analysis is different. This work that we've been doing, and I was involved in for the past 7 years at the Department of Environmental Science, was led by Karsten Sjur-Jakobsen here and Leah Ellegård Jensen. I've put their information if you want to contact them. Otherwise, reach out to Anders and he will point out the right people. That led to also an automated pipeline that we've published on our GitHub page that everyone can use. Very briefly, we start with sequencing all the RNA we can get in a sample. We sort it into 3 big bins. We use this one to get information about 16S and 18S primarily. This is the large subunit of the RNA sequence. At the moment, it's not very informative because the databases are lacking. And then we have the mRNA where we can investigate functions. So what do we do with that? This is an example, and I also put Johanna here because she was the one that made this an R package to analyze the output of our pipeline in an AU color scheme. So if you want, all these are on our GitHub page. You can go find the tools. What you can see is that we get information of the total community, and here we have relative abundances of eukaryotes, of course, at a higher level, but we get a big table that we can go as deep as we want down to species level. It's just that it's very difficult to visualize it. And also we get the prokaryotes down here. Now, when this would look similar to an analysis that we would have done with metagenomic sequencing and amplicon sequencing. And I want to show you this example here because sometimes we look at who is there and then we can say, as you can see here, there's no clear clustering on our analysis, saying that, okay, nothing really happens, the community is stable, functions are still the same. But we do know we have data from the field that something did happen around here that we cannot see on a community level. But if we look at the functions expressed, we can see that suddenly there is a clear clustering that we have also confirmed with statistical analysis. We know that the community rapidly responds to environmental perturbations. Where this in here was a snowfall event that led to freezing on the ice sheet. As I said, the environment still doesn't matter. But once the conditions came back to normal, the community was also rapid to change its metabolic potential. So when we are looking at biodiversity, especially at the microscopic level, where we cannot rely on camera observations or anything else, this kind of work it provides a lot of information that otherwise would be unable to see. Here I have an example where you use a very general database that encompasses many different functions, but if one is interested in carbon cycling or nitrogen cycling, phosphorus cycling, or any other processes, degradation of xenobiotics, there are different databases one can use, or you can make your own databases. And as I said, on our GitHub page, we have tools to help analyze these data sets. Now, for that, I'm going to thank you for your attention, and I'll take any questions that you may have. And back to you, Anas.
Thank you, Thanasis. I hope I'm still. You can still hear me.
Yeah.
Anyway, so we're actually a little bit, I believe, in front time-wise, and but now let's take some questions. So I think that I'll start out with a question for Signe. Are you there?
Yes, I'm here.
Good. And you can maybe show your faces now if you want to, broadly around. So Signe, I was, the message that I get from your presentation is that it's very difficult to kind of to restore biodiversity in agricultural systems, and it's a little bit depressing to me. But is it so that when you have an effect when you in some agrosystems when you kind of make it more sustainable and avoid leaking so many nutrients to our waters? Isn't it so that in this case you will actually see an effect on diversity in our fjords and so on? Is that possible?
Yes, so that is possible. So that was what I tried to say mainly with one sentence, that of course if you change your agricultural production so that you reduce the nutrients impacts on, it can also be phosphorus in lakes and rivers and the fjords. You are of course contributing to assuring the conditions so there's space for restoration and protection. So you are minimizing the pressures, but you're not, and creating conditions for recovery. But the challenge is that because we through so many years have thought that we can solve it within the landscape, the land sharing, we can't. And what we mainly talk about is reducing, but not actually improving.
Okay, so it's a mix of a comment and a question. You gave some figures about the cost of restoring the environment, and I think you mentioned a figure around 6.5 billion per year, plus some extra, and it just strikes me that it's actually not much money. I mean, it's the cost of five fighter planes or so. I mean, when you compare to that, it's incredible that we don't do anything. So how can you work in the council, Biodiversity Council? So what are the politicians' take on this?
I do not know in general what there's many political parties and there's many takes on it. I think that there's slowly an acceptance that we as a country cannot continue to say we are doing it good. We are doing it at least as good as everyone else. There's an increasingly acceptance of that. Yeah, we have not done enough. We need to do more. There have been, through the last year, the main focus on that the green tripartite will basically contribute substantially to solve the biodiversity crisis. And I think at least there is some increasing understanding that there is simply not enough money, and the subsidy schemes and the current the current measures taking are not enough. And of course, I agree with you. It is a little bit depressing. On the other hand, you can say we have simply just not. We have just done the wrong things, and we have just not done enough.
Yeah.
So, but we have the opportunity for relatively few costs to do it better. And then I think it's just a wish for sort of the broader scientific community, is that we tend to just talk about, then we help biodiversity, but we are often not differentiating to which degree we are increasing, because we can increase biodiversity within a agricultural field, or we can at least increase the richness, we can increase the genetic diversity, and we can assure that populations are better connected, that the agricultural system is better functioning. But it doesn't solve the crisis.
No.
So we need to be better talking about what it takes to solve the crisis and actually make a difference. And what we should do at the same time is, of course, also try to make more robust agricultural system, improving soil health. But that is it's for the benefit of us. And it's on a slow trend of giving something back that we have taken through generations.
Advantage
so
of,
it is, you shouldn't stop doing it just because I might say it's a bit depressive, but it is just this, we have just been talking too much about that we will solve it. We can solve it with sharing. We can solve it on the agricultural surface. We are improving. Yeah, you are reducing pressures. But we need the restoration. We need the protection. Yeah.
So okay. Thank you. Yeah. So please raise your hand, and if you have questions, you probably have some questions. So please,
there is
just
a few questions in the chat.
Maybe Morten can take care of the chat. That would be nice.
Yeah, there is one for Signe, that was for your presentation on if it's the same 2% that are protected that is also considered good conditions. Are quite interesting. Is it actually the areas that we protect that we assure through protection is in good condition, or is it different areas?
Yeah, so the answer is actually no, because the analysis is done in a different way, and the 2% is within existing subsidy schemes, and in the Biodiversity Council we are also counting new areas which are not counted. So technically it's not the same. So that is the short answer. But and the main reason why they're not the same answer is this yellow pot of things that needs to be individually evaluated, and which where these 2% are part of. But it's an estimate. It's a gross estimate from the European Commission, and it's not specific areas they're pointing to.
Okay. Please come forward with questions. Raise your hand. Don't be shy.
Kirill, you had a couple of questions in the chat, but maybe it's better if you post them or combine them.
Come forward, Kirill.
Thanks, Morten. Seen a very interesting presentation, and actually quite some new information. So my first thought was, is it those, well, now I'm aware that they are about 2%. Is it those areas that are also bearing the highest potential for increase, or actually for making a difference in improving the biodiversity? Because the rest of the agricultural land, as you said, there is a potential, but you're not sure if it will solve the biodiversity crisis. And my second question is on agroforestry. What do you see? Because I'm from the agronomic sector, agroenvironmental sciences. So what is the potential of agroforestry from your side to solve the biodiversity crisis? You're using this expression in particular, so it's quite interesting. Thank you.
Yeah, so the first question you have is sort of in terms of if those areas which we are pointing to, we have these maps that point to different areas which should be has a focus on the in terms of reverting the loss. They are they are the best current guess on the best analysis until now on where is the highest biodiversity potential and realized biodiversity in Denmark. There is a challenge in terms of just if we would only protect where nature is today and where species are today, we would not cover in 20, 30 years representative all the species in Denmark. So the analysis try to take into account both the realized biodiversity and the biodiversity potential. There's some high productive agricultural soils that is also important for biodiversity, but there's a part of the habitat space we have reduced a lot. Wetlands, some dry meadows, very high, very clay-rich soils, peat-rich soils, which are very important for biodiversity. And if we would just continue saying that we are not giving any high productive land back to nature, we will not be. There are some of these areas that have a high biodiversity potential and also a high agricultural productive value. And then it's of course a societal decision of how do you, what do you value most and what do you take out where. That was a way of answering your first question. The other one is with agroforestry. So it is what you call lock in or set aside is it what it takes? But then on the rest of the surface, agroforestry comes in many different flavors, and some of it I don't see as different than a cornfield with a few more species. But some agroforestry systems, you can increase within those system habitats for tree living organisms, and there is some old cultural systems also in other European countries which are important habitats for a few birds or some amphibians. And so it's not that it is contributing, it is each time you create a bit more nature with quality that actually has a quality and spaces for the species in the long term. So not a flower strip that is gone in 1 or 2 years, but actually habitats that has a long persistence. You are contributing, but not to reverting the loss, but to increasing the functional ecological connectivity. If you read into the large agreement in Europe, they talk about the ecological connectivity. Populations need to be connected. And with initiatives and the right initiatives within the agricultural landscapes, you can contribute to that.
Morten?
Yes. So I have a question that for you, Signe, that couples your point about really the huge need for improving the biodiversity and converting the development of the crisis to this monitoring, and the presentation that Quentin and Thanasis gave, that shows new possibilities in the detailed data and information we can retrieve from different areas and biodiversity schemes, or also not where it's not present. What do you think? This new type of data that is more detailed, maybe we get more conclusions. How can we use that to the best possible way in terms of arguing, your case or the obvious case that we need more biodiversity and we need to solve the crisis? How can we use these new tools and more detailed data?
So I think sort of all the detailed data is extremely valuable for better understanding the system and where all the unseen part of biodiversity are. There's so much we don't know. So we're just getting more insight into the microbial community, the bacterial communities, which worms are in the soil. We better understand where species are distributed. We can also better quantify the biodiversity value of each area and how it complements, how each individual area actually complement to protecting and restoring the total biodiversity in Denmark. So all that value, all these tools are very valuable, and we work on other sounds and also camera traps and with coupling with remote sensing. I think that's very valuable, but it's not an immediate solution because it's very difficult to get mechanisms that are actually paying. If you find 5 more earthworms, do you then get more paid? And you do an action and it takes maybe 2-3 years, you have more microbes, then you get a little bit more plants, but they're still common. It takes 20 years before you have habitats for some of the rare ones, and 100 years before your forests, or even more than 100 years before your forest is actually biodiverse and have the variety of habitats needed. So the financial mechanisms needs to not be on the actual biodiversity gained. I think we have done a report in the Biodiversity Council also where we talk about how within the Danish regulatory mechanisms, what would it take? We would need a system where you can contribute to the general efforts of averting loss in Denmark by protecting and restoring. So you basically so the money flows from private companies and into protection and restoration on the actual actions taken the right places. And then we need, of course, to monitor that they actually also recover biodiversity, because our actions might, given climate change, not be the right one. So then we get into adaptive management of those areas. But the financing need to flow into, to do a difference fast. The financing need to flow into reducing the pressure. So that is changing basically how we grow, what we eat, and so on. But it's also flowing into actually protecting and restoration, and then it can flow up in the ESG reporting and so on for companies. That was a long answer again. Sorry.
Thank you.
Okay, I have myself now. And so I find it so fascinating with the RNA techniques. I mean, we have for a long time restricted measuring of biodiversity to who are they and how many are they and so on. But with the RNA techniques that Thanasis was talking about, then we have an opportunity to know something about what are they actually doing. And but it's restricted to microbial community and life in general. And so I would just ask you, Thanasis, is it possible to, how do you see the years coming? Which organisms groups can be measured meaningful in and also in practice using RNA technology? And I'm also looking at Quentin, you know, because he's working with earthworms. So would that be to link the RNA technology with the remote sensing and imaging technologies that Quentin is working with?
Yeah, that's
an easy question, right?
Yeah, super easy. Yeah, the answer is yes. No. So I think with the RNA techniques, we are limited by how much environmental RNA we're able to detect. For example, with DNA, a new field that has emerged over the past few years is the field of eDNA, environmental DNA, where you can monitor larger organisms by finding their DNA in the environment. That's because it is quite stable. RNA is not stable. So when we perform our analysis, as I said, we start from extracted nucleic acids. There we're limited by the kits. If we collect a large enough sample that we can process, where we have bigger organisms in there, maybe some insects, maybe part of an earthworm that was unlucky to find itself there, we can definitely detect it. Every time we do this analysis, because we collect the sample and flash freeze it, we do find traces of macro eukaryotes also. In all the samples we have analyzed from agricultural soils in Denmark, we do find signatures from the plants themselves. But in a microbial ecology point of view, we're not interested in the plant. We know, for example, that this is a field that we grow wheat on. So we remove that information, but we do have it. So that's it's not a problem of actually getting or analyzing the data. It's a matter of, can we actually collect it to start with, as we do with eDNA. Now with combining remote sensing and sequencing techniques, that's a bit more difficult. This is something that we've started doing, and my colleagues now at the Department of Environmental Science will continue, would be in Greenland, because there you take a satellite image or a drone image of the ice, and that it's clear. You can see if it has a different coloration or not. How do you do that in a soil setup? How do you do that if you have canopy coverage? Never say never, because we've come a long way over the past only few decades, but I don't think it's something that we can do in the next couple of years.
No, I know it was difficult. Signe, and maybe Quentin have a comment on this as well afterwards. Signe?
Yeah, it was just a follow-up comment. So what I completely agree with you, Tanessus, that it's really difficult, but we have done a pilot weekend on Zealand at a big restoration area where we have the eDNA, the soil samples for the microbial communities, and we do the plant communities, and we are then we have had a hyperspectral sensor that came up this summer. There's 3 of these sensors in the universe. There's one on a satellite, then one in NASA, and then one in Switzerland. And this hyperspectral sensor is then mapping the landscape, and we are able to derive characteristics of both N and P and functions of the plant communities, but we can then only start predicting what might be in the soil. So it's always like a prediction. We do not actually know they are there unless we go and sample. So it is, yeah. And I think we are not there before in 10 years of having really good models of the functions and a good model of what is actually in the soil seen from space.
Yep.
I only have a follow-up on Signe's comment. Yeah, so sorry, Quentin. Do you have a follow-up as well?
Yeah, it's no, it's related as well. It's
yeah, okay, go ahead.
Just to second that, yeah, I think combining methods is actually the way forward. So there is no way we could just use images or just use remote sensing or just use molecular methods. What seems to be working better and better now is like we realize we cannot do everything with images, so we try to take DNA to make a species list, and then we use that to match the images we have to the known species list. Because yeah, with millions and millions of insects, you can't just hope to do everything with images.
No.
Yeah.
Tanessus.
And what I wanted to say is that I gave an example that we try to combine remote sensing with sequencing in Greenland, on the Greenland ice sheet specifically, because of course we don't have any plant growth there, but also the communities there, the microbial communities, are quite limited in diversity. When you take a soil sample and we do the analysis we find 10, 11, 12,000 taxa. So of course the complexity is much higher. We cannot just predict who is there by using other measures. But on the ice sheet, things are much simpler, even though we still have a lot to learn, and that's why we try that. But yeah, I agree with you. It's and I'm quite happy to hear that more cameras, more advanced solutions are coming into play.
So it takes me to one of the questions in the chat. That's a question. It relates to this, I believe. When do you think there is a monitoring setup that is affordable and useful for the average farmer who wants to monitor at farm level? That's an easy one too.
Is that for me?
Yeah.
Yeah, so for farm, it's a little bit of a different operations because often farmers have a financial incentive. So if they see a benefit of monitoring a very specific group of insects, for instance, say pests, then they will do it. And it's what a lot of the community has been doing. So there are countless companies that set up smart insect traps in the field. And they're often a little bit different from more agnostic biodiversity monitoring because they sometimes use an attractant to target one pest and they have a system basically tuned to this one pest. And they try to promote more of an alert system. So you have a detection of a pest and then it sends you a text and it says it's around. It's not really shown to always work. Sometimes it's actually counterproductive because you have alerts that are a little bit false positive. Like there is a farmer 20 kilometers away that has spotted one pest, then you should spray now. So it's not so far off for farms, but they have more money. And because they're often in monoculture, the environment is quite homogeneous. So you don't need so much spatial coverage. You can put 2 or 3 traps on your farm, and that's maybe enough for you.
I come to think about that with your approach, Quentin. I mean, what you say, your resolution in time is quite high because from hour to hour it would be possible to measure insects hitting your plate and so on. But so that's difficult with the DNA and especially RNA technology because, I mean, an online approach is, I mean, when you can read about Oxford nanopore that can be used in an online real-time, I mean, approach, but this is not really real-time, isn't it? Or you still need to go to the side and you need to extract some DNA and then you need to have a small fungal or nanopore device that is portable in order to, right?
Yeah, that's correct. I mean, with nanopore, you can start analyzing your data more or less as soon as you generate it, but you have to collect the sample, you have to extract the DNA, you have to prepare the library.
Yeah.
So
when will we have automated DNA extraction?
Well, Nanopore did have a product before. It was called the Voltrax. They discontinued it. I don't know if they have anything to replace it.
Thank you. Any more questions, please? We are also getting close to the end of the webinar, but we have room for a few more questions if you have, or comments. Okay. Seems not to be the question. Morten.
Yeah, I have a comment, and maybe it's a comment with question with that can be commented, but I really think that it would be of huge benefit that we in the sort of circular bioeconomy community start and many of us already started, but really incorporate this idea and the actual need for converting the biodiversity crisis and improving that. And it requires to set aside, it has a lot to do with land use and many of the things we are working on in sort of circular bioeconomy. Well, it requires bioresources, but it also suggests to change the current agricultural system to a large extent and also make technologies that require much less area and also resources from the seas, using the bioresources several times, etc. But it would be really good to think about how we then incorporate that argument that we really need more land area where we have biodiversity full on, and applying those arguments, I think, is really good, and applying them in the realistic needs that Signe presented. I think that is really important, that we do that. So it was just a comment and reflection from my side, but thank you very much to all presenters, and yeah, if anyone wants to comment that, welcome.
Well, I just you just, I think, make a nice roundup of what Signe said, and what I took in from Signe's presentation was also that we need really to dedicate some land for nature to have on its live on its own premises and develop freely biodiversity. And we will touch on the impact of diversification in agrosystem on biodiversity in the next webinar. So, and I also got a message from Thanasis and Quentin that there are some exciting new technologies that are kind of in the infancy, but with very good prospects for with time to kind of to give us more data on diversity, and not only, what should I say, species diversity, but also function and behavior. There's room for one final catch up or comment. Otherwise, Stine. I know what you will say, Stine.
Yeah. Hi, Anders. It's just because I want to mention the survey that we have prepared.
Exactly.
Yeah. Thank you.
And
if you will show the slide.
I will. No. So what Stine urged me to tell is that there is yeah, okay, then I need to actually share my screen again, and I'll just let it. Can you see my screen now? There is a QR code in the rightmost low corner where you can use it and fill in a small short survey about the webinar here, and maybe also give some comments on how we can improve it, and some good ideas. And then finally, I would say that I would urge you to go to our webpage and see what happens in CBIO, and also become a member of CBIO. So do that. It's also possible for external to participate as an external partner. Okay, then I'll say goodbye, but leave my presentation open here so you have time to use a QR code. Thank you, and thank you so much, especially to the speakers, and not so much thank you for the EU IT department that closed me down. Yeah, Hubert?
That was a mistake. I just wanted to applaud.